Welcome to The Machine, a podcast from Waterford Institute of Technology. My name is Rob O'Connor. I'm a lecturer in the Department of Computing and Mathematics at WIT. You can find us on Spotify, Apple Podcasts, Pocket Casts, Anchor, Castbox, Overcast, or pretty much anywhere you can find your podcasts. We're also on social media at machine_podcast on Twitter, and a very unloved Facebook page, Machine Podcasts, just because I'm trying to avoid as much Facebook as possible at the moment for my own sanity. In the last episode, we discussed some research WIT is involved with through the TSSG Research Group, focusing on integrating COVID-19 assistance technology with society, clinical data as a software service, blockchain-based certification of public health compliance, ensuring social distancing through low-cost wearable tech, and others. That's available now if you want to go and listen to it. But today we are going to discuss some COVID-19 treatment research, specifically two projects, treating lung tissue damage due to COVID-19, and also molecular communication modelling of COVID-19 in the respiratory system. Now, whatever about the technical areas, I'm really out of my depth here, but thankfully we have a number of people who know their stuff to explain it all to us. We have Dixon Vimalajeewa, Bruna Fonesca, Sasitharan Balasubramaniam, all researchers in TSSG, and maybe we could get a quick introduction from each of you, what your role is, where you are, and what your normal day-to-day kind of activities are. So Dixon, we might start with yourself.
Hi, my name is Dixon Vimalajeewa, and I'm a postdoctoral doctoral researcher in TSSG. And basically, I'm currently working on mainly on the COVID-19 project. And on my day-to-day work, I just apart from this COVID-19 project, I work on how to personalize milk consumption, and that's what I do these days.
So you're coming at things largely from a kind of an agri-tech background, is it?
Yeah. Basically, my background is like mathematics and statistics, but right now I'm working in the agri sector, how to use the machine learning and data analytics in agri applications side.
Brilliant. And Bruna, could you get a quick introduction from yourself?
Oh, yeah, sure. Okay, hi guys, I'm Bruna, and I'm a PhD student at TSSG. I'm an electrical engineer. I'm from Brazil, and actually I'm working on the bio-nano device areas for treating diseases. And my day by day at home is just working on the COVID research, and sometimes watching the river in front of my house and see the swans catching the food. That's funny.
Brilliant. And Sasi, apologies for mangling your name. I've been doing that now for about 10 years and I probably never get it right. But we have Sasitharan Balasubramaniam. Did I get it correct there, Sasi?
That's perfect, Rob. Yes.
You're very kind. Sasi, you might just give us a kind of a brief overview as to what your role is and all the pies that you have fingers in.
So I'm the director of research at the TSSG right now. I'm co-directing it with my colleague Kevin Doolin, who's the director of innovation of TSSG. I took on this role about 3 years ago. So my main focus is to focus on the research, the basic research of the TSSG, as well as some of the applied research, while Kevin is focused on more on the applied and the commercialization research. So we look at the TSSG, we look at various different types of research in ICT and in particular interdisciplinary research, not only on the fundamentals of ICT and in particular for communication technologies, but how can they be applied to healthcare, in agri-tech, in energy, and also in transport as well. So we're looking at conventional techniques of telecoms that we know every day onto really new emerging techniques of actually communicating, which we're going to be talking about today.
Okay, so the two projects we're going to discuss today, treating lung tissue damage due to COVID-19, and Bruna, that's your project, you're involved directly in that, and molecular communication modelling of COVID-19 in the respiratory system, and Dixon is directly involved in that, and Sasi's involved in both of them. Sure, perhaps Sasi, you might give us a kind of a brief overview as to what each project is, just to kind of set the scene, if that's okay.
Sure, sure. So just to give a little bit of background, in the TSSG a good few years ago, we started this new area of work called the Internet of Nano Things. So we all know what the Internet of Things is, where all your smart devices are all connected to the internet, effectively feeding all important information to the internet. But we came up with the concept, what if we can actually miniaturize these devices into very, very small devices constructed out of nanoscale components? And can they actually feed information from areas we may not usually be able to go deep into? Let's say, for example, deep in our tissues, in our lungs, feeding this information up to some healthcare record we might have on the internet. So there's a brand new area that came up, I would say maybe about a good 15 years ago, called molecular communications. So when we think of communications, we always think of wireless. A very good example is your mobile phone that communicates using electromagnetic waves. But at the same time, there's another form of communication that's been around for billions of years. And a very good example is inside a human body. And they actually use molecules to communicate with each other. So the way the brain, say for example, the brain talks to the different organs, or even the way the different organs are talking to each other to self-regulate your human body, is all by molecules. So we figured that, can we extract information that is communicated through using these molecules? And can we also construct artificial devices that use molecules for communication?
So can I just clarify something there with you, Sasi? So are you talking about the way your brain would send a chemical signal to an organ to do some work, or equally send another type of signal to a muscle or exactly. to whatever? Is it? Yeah. Okay. So exactly. It's looking at that form of communication.
Exactly. It is like as if your laptop is communicating to your Wi-Fi. There is a channel, and then it's actually sending electromagnetic waves. In this particular case, it's all chemical messaging that's actually being sent from, say, like the example you said, Rob, from organs going up to the brain itself. So we use the typical telecommunication techniques, the protocols that's actually out there. We map it now to a biological system. So we have a number of topics in that area in the TSSG. We do things like developing biological computers using our cells based on engineering their communication. You get like AND gates, OR gates, like your digital system, so that you can implant them into, deep engineer the cells to actually be a gate. And you might have them sitting in the gut, and they can compute the different enzymes to find out whether you have a disease of a particular type or not.
So again, to stop you there, Sasi, because I don't really know much about this, so you'll excuse my ignorance. So are you actually saying that there's a kind of a binary logic-based system built using cell technology, or using biological cells that can then perform logical reasoning?
That's a very good question. In fact, the cells actually communicate analog signals, but they have a set threshold where they rise from low to very high with that analog signal. So we can interpret that like as if it's a binary signal.
Right. So in the same way there's an electrical signal, we say something is on or off. It's not really, it's kind of low or high. And when it goes exactly. above or below a certain threshold, we say that. And it's like that, but in a biological context. Is that it?
Exactly. Yes.
Wow.
Yeah. So now when you have these molecular communication systems inside the body, the big question is, how do you control it from the outside? How do you use some external device to actually control them and then send commands to them to trigger or do certain types of functions? So can I go into the details of Bruna and Dixon's work, just to give a bit of detail into that now?
Absolutely, please do.
So
yes.
Coming from molecular communication perspective, what Dixon is doing is trying to model how the virus travels through the lungs using molecular communication paradigm. So in this particular case, the transmitter would be essentially the input into your nose or your mouth, where the virus actually goes through. And the channel will effectively be your airway. And the receiver will be the endpoints wherever the virus binds to. So by modeling this as a communication system, you can then find out what of these viruses as they're going through. Is there an amplification point when the virus binds and replicates? Is there a filtering point that we might give this as an indication to drug designers to develop new drugs to point certain areas to say, hey, that's an area you want to trap as much virus before it goes deep?
So is the idea to get a detailed analysis of the roadmap and the life cycle of the virus throughout the human body and the human system. Is that correct?
So for in the case that what we're doing for COVID, we're only focused in the lung airway at the moment.
Yeah. Okay.
So from the point it entry, it goes in to the point it flows through the lungs itself, up to the point it binds to some cell and actually infects that cells inside.
Okay. Excellent.
So we want to know, like, say for example, people with different breathing rate, how that impacts on the way it actually pushes the virus down. Is there different breathing rates between young people and old people? Is there different breathing rates between smokers and non-smokers? And the thing with COVID is that it actually binds to cells that actually express what they call ACE2 on the surface of the cell. If any cell has that expression on the surface, the virus can bind to it easily. So if you don't have that AC expression on that particular cell, the virus effectively just bounces off it. So now there has been study done, I think Dixon can explain this better, that smokers tend to have cells that express more ACE. So that's why the virus might actually bind much more easily into patients who have those. So in a simulation model that Dixon has created, we can change the quantity of ACE along the airway. And then if you have an X quantity of virus going in, you can find out, hey, what's the difference between a smoker and a non-smoker? Where does the virus travel through, replicate, and propagate down the lungs? And this type of model will be very, very important for drug designers in particular. If they want to target certain areas, the lungs or the airways, where should they do for different people?
Brilliant. And do you want to give us an overview of Bruna's project, treating lung tissue damage?
Sure. Exactly. So what actually then happens is that if in case so we collaborate with Gerald Barry at UCD, he's the one who's actually giving us all the knowledge about what COVID is. We're actually not biologists ourselves, So through our collaboration, we found out that some of these viruses, when it goes deep down, when it actually infects the cells, it can actually damage the cells and actually create a lot of damage inside the lungs. And some patients actually, end up, dying from that effect as well, in the case that the immune system is not able to actually fix this up in time. So we found out that in China, there was some study done on actually using stem cells to actually help to actually repair the lung damage that results from COVID infection. So we thought, okay, can we come up with a concept where we can find a way of delivering stem cells through the airway down into the lungs, and that they will sit in there, and from outside we can control it so that they can be released, and in order to actually repair the lung damage. So what Bruna is actually looking at is another form of molecular communication where she encapsulates these stem cells into little polymer sacs. And you can push this through a ventilator. The patient sucks it in, it goes down the airway. And from the outside, you can use ultrasound waves to actually emit. When the ultrasound waves goes in, it breaks the polymer and releases the stem cells. So you can actually dictate the areas you actually want to release it. So in the case where patients who have been cured from COVID, there are chances the virus has come back. We've seen some studies on that before. So what you can actually have is you can have these sacs sitting there, and in the event that the virus does come back, all you do is just put an ultrasound transducer on the outside, send the waves in, break the sacs, and then they'll help repair the lung damage before the damage is just too high for the patient.
Well, we might go to Bruna then at this point, if that's okay, and to just get a bit more detail. So Bruna, specifically, what area are you directly involved in this treating lung tissue damage with COVID-19 project?
Okay, so before the COVID-19 research, I was working with electrical stimulation with bacteria. And one of the approaches that I used for the stimulation was the use of ultrasound stimulations. And when we were thinking on how to use that technology that I was trying to use for the use of COVID-19, to the research with COVID-19, we are thinking, okay, so maybe we can use the ultrasound to break the nano devices, because these nano devices are made of polymers that are sensible to ultrasound. So when the ultrasound waves passing through the polymer surface, it breaks and release the stem cells. So the what I'm doing is just using what I was searching for, the bacterial research with ultrasound simulation, for the ultrasound simulation of the nano device in the project.
And this project is not a solo run. There is a collaboration here. There's Rochester Institute of Technology in New York in the USA, and also the University of Tampere in Finland, as well as some of our more lab-based colleagues in WIT through the PMBRC research group. It's an interesting confluence of disciplines.
Yeah.
How have you found jumping between all of these horses, if you know what I mean?
It was really difficult because I had to catch up with all the concepts that are involved with the research. So I'm learning a little bit of everything in one time. So it's been a little bit challenging for me.
Yeah, I'd imagine it is, and also now that we're working in this remote type of environment where you can't sit down and have a cup of coffee and a chat with somebody, does that make it more difficult?
No, it makes more simple the conversation, especially in this situation because we are at home. So we're having more meetings, Skype meetings, so we can discuss more about what we are doing and the concepts that we can improve for the research. So it's been a little bit easier to learn more the concepts.
Excellent. Dixon, we might go to yourself now if that's okay. So again, talking about molecular communication modelling of COVID-19 in the respiratory system, which again, right now I'm kind of ignorant about this type of stuff, so you'll excuse my stupid questions and I'm going to ask you. So with this modelling, I mean, Sasi talked about how this type of model could be interesting for drug manufacturers or for testing. What is the kind of the threshold that you can run what if simulations with your model? Can you kind of more or less test anything?
But based on different physical parameters such as age and what is our day-to-day behavior, such as like smoking and underlying health condition, how those factors could impact the severity or infectivity of SARS-CoV. So if we know, like suppose for example, if somebody smoke and how that person will get severe condition, or whether he have mild condition, or how long it will take to convert into severe condition, something like that. We can characterize characterize the infectivity of COVID-19 based on our physical parameters and as well as biological parameters.
So not trying to stay away from COVID-19 because that's obviously the focus of the conversation, but I would imagine that this type of approach is applicable to pretty much any sort of a virus or any sort of infection where you can get measurement information. Would I be correct in saying that?
Yeah, that's correct. Yeah, if we can model the virus infection process, so it's not only in at the moment we are considering only the respiratory system, but the virus, any kind of virus, for example. So the COVID could also infect our heart or in our intestine or like our kidneys. So how we can explain the infection in, for example, in the heart, how it progresses, what is the initial, how virus could propagate up to the heart, and what kind of infection could take place in the heart, and what is the severity of the infection, how long it will take, and what are the parameters which could affect the infectivity, like heart failure or any kind of high blood pressure. How we can take into consideration those factors, and how those, if we can explain those such things, we can easily target our treatments.
And I'd imagine the from a purely kind of computational power point of view, this is, I'd imagine this is quite significant. I mean, you're going to be dealing with a massive amount of data and a massive frequency of data.
Yeah.
So how are you able to manage that when you're at home?
Basically, initially I start with the simulated data, and then I try to get some like a realistic output from my model. Then I just first of all I want to make sure that my model works well. I can characterize, I can explain some real world scenarios, like suppose like COVID infection. Then I should be able to compare with the existing and already published results. Once I confirm with that, then I I get some data I search what is going on with COVID-19, what kind of data people have published, and how can I use such data, suppose, for example, what is the replication rate of COVID-19, what are the most, how people have measured the infectivity, and what is the initial how initial exposure could impact the virus infectivity. Such kind of I try to find such data from the recent publications related to COVID-19, and then I use that data. Anyways, that finding data is really difficult at the moment, because nobody knows much about COVID-19. So people say, like, already there are some conflicts. Sometimes people say, okay, this factor works well, but some people say, no, it doesn't work, because we don't have enough data to confirm that. So I face such kind of difficulties, and I want to find the data which I want. And at the moment, it's very difficult to find data. That's the main challenge.
And are there, I mean, I hear about international collaboration. I mean, the project we were just talking about with Bruna is the perfect example of that. I know you're working with Teagasc here in Ireland on this, but is there any sort of an international effort to get, if I called it a standard data set, I'm probably doing it a disservice, but I think you might know what I mean, on COVID-19, so that everyone can at least be working from the same starting point?
Yeah, but yeah, that's I agree. But the issue is people keep publishing data, but there's no sufficient data. Suppose in my case, I just need exact values. Suppose, like, what is the replication rate? So people cannot justify, okay, this is the standard replication rate, this is the infectivity, because there's no enough data. The clinical trials are still going on, and people keep changing the values. Okay, suppose in China they published, suppose, for example, the replication rate based on their data, but if you consider Germany or somewhere, they published some other data. So, like, if I talk about replication, so the data is not like, is still not ready to define, okay, this is the standard value for this thing. For suppose, still we don't know the density of COVID-19 virus particle. So we consider the COVID-19 virus particles are similar to SARS-CoV, but that's how we consider in our simulation. But still we don't know what is the standard value. So it's a kind of challenge for us when I'm doing the simulation.
So what you're saying is, and again, if I'm incorrect, please do let me know, is that you're making certain assumptions in your model based on the data that's available to you, but you're not, I know you can never get 100% certainty, right? Because I know that's not how these things work. But you're basically saying that if you had more data, you would be able to get a more accurate model. But that data doesn't actually exist yet. Is that correct?
Some data is not exist yet, but I try in my best to use all real values instead of like making assumption, because it helps me to get more realistic outcomes. Instead of just making a lot of assumptions, I always try to use more real values. So that's what I'm doing. Yeah.
Well, Sasi, maybe we might go back to yourself on that then, because talking about the international effort in COVID-19, What Dixon has described there would sound like a kind of a basic building block for any sort of international effort. Am I correct in that, or am I missing something?
Yeah, you are. Like, especially within the EU, we've started to see various different, let's say, for example, research calls come out for people to sub proposals in to address this so that we can generate more data, crucial data, find a way to link from, let's say, for example, patients in the hospital with raw data that they've got, clinical data, to actually allow us to actually take that data and make some use out of it. But just to add on to that, Rob, like say for example, right now we are part of a proposal that's being led by University of Oslo for EU H2020 Future Emerging Technologies (FET) project, where we want to develop what we call a human on a chip. So you get different organs that you can grow from a particular human, say a kidney, lung, hook them all up through blood vessels on a chip. And then we want to find out how the virus is actually infecting those organs and how they're actually propagating between the different organs as well. See, that type of data would be literally very crucial for our studies and other studies as well, because you go down to the cell level and it's outside the body. And you can actually take that then and there, very different types of parameters.
So just to clarify, what you're talking about there is a virtual human, not a real person. It's
exactly, yeah. In fact, we call it a biological avatar.
Yeah.
So human on a chip is not something new, though. The WIT Institute at Harvard has been developing this. So you get these little chips, and you can actually place lung tissue in it, or it will have everything in it: the epithelial cells, the blood vessels, everything. It's all there. It's as if it's a piece of your lung itself, And they've hooked it all up to the kidney, they've now got a mini brain as well. They call it brain organoids as well. And you can actually watch how the virus is infecting and how it's even migrating through the circulatory system, or even through some are now saying it might even migrate to the nervous system as well.
Yeah, I heard about that. They're saying they can go into the brain. That's
Exactly.
quite scary.
It is, yeah. I'm still unsure how it actually goes. They say it's through the nasal passage, but now how does it go from there right up into the brain, cross the blood-brain barrier to the neurons? I don't know. It's a big mystery.
But again, when you describe this human on a chip, that I won't say it'll solve it, but it might help to unlock that mystery.
It would, and also what we're hoping to do is that you might be able to put as well in such a way that if a patient X comes in, you take their genetic data out and create this virtual avatar of that particular human, and you can then find out if they have all these ACE cells with ACE expression, how the replication rate is different from another patient. So you might be able to tailor the treatment specifically to that patient.
Wow, fascinating. I mean, again, what's going across these projects and even the projects that we were discussing last week, which were more technologically focused, but again, that interdisciplinary ethos that's taking place here, and it's not just with yourselves, this is all across the world. It is a difficult balancing act, because you're describing communications technology, computational technology, mathematics, molecular biology, immunology, virology, all sorts of different areas that on anyone on their own is a significant amount of knowledge for a single human to have.
Yeah, you're right, Rob. I mean, the reason for the driving force of bringing different disciplines in is because the complexity of the problems we're having to tackle nowadays. If you were looking to look, let's say 20, 30 years ago, a lot of the different problems, each discipline might have been able to cope with it. It's not a problem. But then suddenly today you're getting viruses that emerge very quickly, spread very quickly as well. Thank God Ebola was able to be contained quite quickly. That was very good. But look at what has happened in this particular case, the SARS virus, So you're very right. And how do we tackle that? Maybe the immunologists and virologists may be able to do it, but what about the speed and time to actually come up with a solution? So our mentality here is that if we bring all these different disciplines in, maybe we can shorten the time of producing solutions. And it could be a different way of producing solution compared to the conventional techniques as well.
So again, whilst we're all socially distancing and isolating and et cetera, it's never been more important for us to collaborate and communicate effectively.
Absolutely. Yeah. You'd be surprised. Like, we're all communication. You yourself, you're a computing person, communication, that technology was your background, but you'd be surprised how the knowledge and the concepts we know is so valuable to people who are in biology, for example, how they might use concepts we might develop for protocols of device to communicate over wireless signals, how that could be used to understand how cells and virus communicate with each other, how cells propagate and move along. Yeah.
So let's, even leaving COVID-19 aside,
Sure.
into the future. I mean, I have a kind of a burst of activity going on in my mind about, wow, all of these things could be happening. I can imagine applications and research areas that are interdisciplinary. I mean, the whole molecular computing thing is mental. I just think about kind of like self-healing systems.
Yes.
I mean, I know that's been going around for quite some time, but like now it sounds to me like it's an actual reality. I'm going to talk about self-healing systems. I'm talking about a system that, if it breaks for some reason, it might actually be able to repair itself without the need of an engineer to come in and physically fix something. Am I right in that?
You are to a certain level. It's actually, you are very right, because, say, for example, the concept of molecular computing is that you sense what are the harmful molecules that might have been emitted by the disease cells, pathogens, compute, and then produce the molecules to actually kill them immediately. So rather than wait for all those markers to travel into the circulatory system, infect all the organs, and you realize you're sick, you go to a doctor, do a blood test. With this concept, you can sense the disease then and there, treat it then and there, at a very, very early stage. So that's the whole idea, and that you might minimize the stress on the healthcare system as well. Because at this point in time, whenever we're sick, we all look in one direction, and that is to the doctors and the clinics and maybe the hospitals as well. Can we take some of that stress out? If some of the diseases could be autonomously treated, but anything that's more severe may need surgical procedures, then that's where you might head to the doctors to get things done.
Yeah. It sounds like science fiction, but I suppose, again, 30 years ago, we're all walking around with phones in our pocket that were on Star Trek, back in the day. And now every kid in Ireland has one, and across the world, all across the place. It's amazing.
You're very right. Actually, that's a very good example you gave there. Is that, exactly when Star Trek, I can't remember who it was in Star Trek, the character who actually had the
Captain Kirk and Spock in the 60s. Yeah.
In the 90s, Motorola created the exact same flip phone, and it became a reality. Now we all take it for granted. We all have it in our pockets, right? So I think the more we can look towards science fiction to get inspirations, I think it's actually a great way to actually come up with new solutions to tackle these really challenging and dynamic problems that's coming up every day.
That's fascinating. Guys, on a totally separate note, how are you all managing personally with the COVID-19? Are you going off your heads? Because what I found is that working here, it's grand, but it takes me 3 times as long to do anything. Maybe because I'm constantly interrupted by small people. How is it for you guys? Are you able to, or perhaps you're working more efficiently that you're at home?
Well, in my case, the problem is that the meetings takes a phenomenal amount of times. I find that, I'm in meetings the whole entire day. Real work only starts after 5 p.m. But yeah, it's different and it's new. But I find a lot of people are similar to me as well, with all the meetings, can really drain you as well. But yeah, it'd be nice to meet people, go back and meet people face to face and talk to them and do work, in the normal conventional way. Can't wait for that.
And Dixon, how about yourself?
In my case, because I'm mostly dealing with mathematics, so I need I need to work continuously, maybe 2-3 hours together, because I need to link everything together. So in that case, I prefer working here because I don't get much disturbance, and so it's helpful. But sometimes, when I'm stuck, I just need to talk to people. For example, if I have to talk to Sasi, instead of having online call, if I can explain things by using a paper, writing down, I know that is much efficient. But anyways, that is challenging. So some point it's good to work like this based on my work, based on the work that I'm doing. But sometimes it's quite challenging. Anyways, somehow I manage both maybe I would say I'm getting used to this.
Yeah. And Bruna, how about yourself?
For me, it's been challenging because, we're at home and we have a lot of distractions. So it's hard to be focused here. And sometimes with the swans fighting against each other in front of my house, it's pretty funny and a little bit distractive. So, but I'm working hard, and I think that I'm a little bit more concentrated in my research right now because I have more time to concentrate in each specific concepts and integrated them in a real the whole concept of COVID-19. And also, we are dealing with the distant with a lot of Skype meetings, so you can catch up with each other easily, and we can say what we're doing, how it is our progress, and everything we can do. Right now, it's been a little bit challenging because we can't do experimental tests with our research because all the labs are closed. So we're trying to focus in the theoretical model and simulations. So it's hard to maintain only the research focus in this theoretical proposition. But we're doing the best, and we're safe we are safe at home. So it's more important to be at home and be safe.
Well, I'd like to thank all of you for your efforts. Fair play to you, and for keeping on going as well. It's not easy, certainly. I find it's not easy working at home. Some ways it is, other ways it's not. So I can only imagine working on complex issues like E. It's challenging, and I think you've all tried to downplay that. And fair play to you. You're mighty individuals. Hopefully we might all get a chance to meet up and maybe carry on this conversation in person at some stage in the relatively near future. But for now, I'd like to thank Dixon, Sasi and Bruna for their time today. And I hope you have a good weekend. I was going to say you're going to do anything nice, go anywhere nice, but sure, we know what's happening there. Look, thanks very much, guys. Stay safe, and we'll hopefully talk to you in person soon.
Great. Thank you, Rob. Thank you very much.
Thank you. Bye-bye.